Hello everybody, welcome to Science Chomp. Let's talk about genetic engineering. Now, in case you don't know, genetic engineering is when you alter the genes of an organism so it becomes modified in some way and it's useful for humans. So let's see how it works. First of all, on the side, what we have is some human DNA and we've identified a gene, and that gene codes for insulin. Most of our bodies produce insulin. If you don't, you're diabetic. So what insulin does, it helps your cells absorb glucose. So if you're diabetic, you don't produce insulin, and we've got to find a way to produce it for you. Previously, before genetic engineering, we just took insulin from another animal and put it into injections and injected people with it. But now we can modify bacteria. So let's see how it works. On the side over here, you got some bacterium and you got your bacterial DNA, but they also have these things called plasmids. Now, in case you don't know, a plasmid is a circular piece of single-stranded DNA that replicates independently of the host cell chromosome, which is that bit over there. So how does it all work? Well, what we've got to do, we've got to take this plasmid and modify it. So this is our plasmid, and what we've got to do is we've got to cut it open using a restriction enzyme. Now, the reason why it's a little bit like this is these things are called sticky ends, and sticky ends will help stick the new gene into this space. So our new gene is actually a little bit like this, and the sticky ends are complementary, so they can stick in there quite nicely, but you still need an enzyme called ligase to stick it all together. So the end product is a bit like this. And then what you do is you inject this back into the bacterium like so, and then your bacterium, your genetically modified bacterium, can now go ahead and produce some insulin. Once it's started to produce insulin, you can basically allow it to replicate inside these giant vats and things, and then just put that insulin into an injection, and voila, you have some medicine. We can also genetically engineer plants. Okay, so this diagram kind of demonstrates exactly what goes on. We don't need to worry about all of it. Let's just think about the basics. You've got this bacterium, Agrobacterium. Normally this bacterium, what it does, it causes like plant tumors, but it's got a plasmid, and suppose we've genetically modified this plasmid over here. What we then do is we can stick it with an embryo and it infects the embryo. It's got a fairly high failure rate, so we have to select which ones have actually worked. And we do that by growing here and then testing them. So this is a culture medium. Once we've got some successful growth, we can just plant them in some soil. And then there you go, you've got your genetically modified plant. And then that plant itself, you can let it reproduce on its own in the natural way, and that will work. Now, there are advantages and disadvantages to genetically modified organisms, GMOs we can call them. Let's look at the advantages first. So generally you get a better crop yield, you get things like disease resistance, you get things like pest resistance, so they can actually produce chemicals which things like caterpillars don't like. We have the ability to grow in poor soil. We have longer shelf life, improved nutritional quality. Because they're bigger, they're generally cheaper, and they emit less carbon, smaller carbon footprint, because you need less land. There we are, they're the advantages. Unfortunately, there are some disadvantages as well. So the disadvantages, there could be long-term health issues. They've been around for a while, genetically modified organisms. Trouble is they haven't been out for that long. Like, it's not been like 30, 40 years, for example. So we don't know the long-term health problems that it could cause with people. The other thing is they can limit biodiversity, and that's bad because the more biodiverse life is, the better. What they basically do is they outcompete natural wild type organisms. Talking about wildlife, it can impact wildlife because a lot of them are basically poisonous to animals. So it impacts wildlife. And think about it, if it's poisonous to things like insects and things because of the pest resistance, well, what kind of effect is it going to have on us? The other thing is, like, if you buy fruits and vegetables from the shop, they don't always label genetically modified organisms. So you might be unwittingly eating them, and that's obviously not a good thing. Also, cross-pollination can happen with wild type organisms, and that is going to basically produce things like super weeds. So these are like weeds, these are plants which you don't want growing in particular areas, and you can't get rid of them because they are basically cross-pollinated with genetically modified super genes, basically. There we are, they're the disadvantages. So it's not all sunshine and roses here. Some of it is cause for concern. As for genetically modified animals, we can do them. Just as before, what you've got to have is you've got to have an embryo, and you've got to inject some genes into the embryo. Again, it's a super high failure rate, so we generally don't tend to do them. But we are experimenting them so that maybe we can grow organs inside of sheep and transplant them into people and this type of thing. So there is use there, but generally it's just restricted to the lab. Also, you can say modify things like cows to produce like particular proteins in their milk and stuff like that. But again, you're talking about these sorts of disadvantages which can come about. So yeah, there we are. Thank you very much for watching, and goodbye.